Hello, I'm Bill MacConnell. MacConnell Research develops instruments and supplies for molecular biology research. This video will illustrate the use of the MiniPrep 24 and 96 instruments. If, after viewing the video, you still have questions, please call us at 1-800-466-7949 or visit our website at MacConnell.com. Doing mini-preps by hand is a tedious process that consumes much laboratory time. That's why we're so excited about the MiniPrep 24 and 96 instruments. These compact yet rugged benchtop instruments purify plasmid DNA automatically. The MiniPrep 24 and 96 instruments will save countless hours of laboratory time. These instruments produce highly pure DNA, which can be used in nearly all molecular biology protocols, including fluorescent automated sequencing, cell transfection, restriction digest, and transcription. These are the MiniPrep 24 and 96 instruments. They differ in the number of samples they can process in one run. The 24 instrument can process up to 48 samples in a little over 1 hour, whereas the MiniPrep 96 can process up to 96 samples in that same amount of time. Operation and setup of the instruments is virtually the same, therefore I will refer to them interchangeably throughout this video. First, we will review features of the instrument. It is very important to thoroughly read and understand the instruction manual prior to beginning. This is one product where the instruction manual is well worth reading. The manual contains many easy-to-understand instructions and illustrations. If, however, you still have questions, please call our technical support department at 1-800-466-7949. I'd like to point out the features of this instrument, starting from the outside and moving to the inside. First, there is the touchpad. Inside the instrument's chamber is an electrophoresis processing rig. This rig has several important features. First are holder slots for the cassettes. The 24-well instrument has 2 slots, while the 96-well instrument has 4. There are electrode leads here, here, and here. And there is a tube that fills and drains the rig. The rig can be disconnected from the instrument, however it is rarely necessary to do so. There is also a removable rig filter here and here. These rig filters should be in place at all times when running the instrument. You can clean the rig filters by simply rinsing them in hot tap water. There are 2 camshafts with cams. These cams automatically rotate once during the instrument's purification cycle. Their rotating motion is what pushes the cassette elution chambers into place at the correct time during the run. This process will be explained later. There is a float switch that senses the correct buffer level during filling. There is also a sensor next to this float switch that reads the buffer temperature. The running buffer temperature and current of the instrument is continuously displayed during the run. The lid switches here and here tell the instrument's computer when the lid is open or closed. They also prevent the possibility of electric shock to the operator, as the lid open condition completely deadens the electrophoretic voltage. Now let's look at the rear panel components. The on-off switch is easy to see. On the left side, there are 2 lengths of tubing that emerge from the rear panel of the instrument. One is for new or unused buffer that is to be placed in the new buffer reservoir. The second tubing, or used buffer tubing, is placed in the discard reservoir. Alternatively, the used buffer tubing can be placed into a drain. In either event, however, the end of the discard tubing should never be lower than the bench surface. There is a third tubing that emerges from the back panel. This is merely an overflow tubing and can be left sitting on the bench. I will now describe setting up the instrument. The large box that you receive that contains the instrument should also contain the instruction manual, power cord, and extra rig filters. A second or additional boxes should contain the cassettes, applicator combs, pill deliverers, or lysis buffer. It will also contain the test culture, 100x running buffer, and growth media. It's very important to store the cassettes at 4 degrees centigrade in a refrigerator. Store the applicator combs or lysis buffers, pill deliverers, and test culture at -20 degrees centigrade. All other components can be stored at room temperature. A clean 2-liter Erlenmeyer flask from your lab should be used as the new buffer reservoir. This flask does not need to be autoclaved, but should be clean at the beginning of each run. A similar flask should be used for the used buffer reservoir. Prior to the first run of each day, you must run a 2% bleach solution through the instrument, followed by a rinse with deionized water. You can make this 2% bleach solution by simply diluting 15 to 20 mils of household bleach into 1 liter of warm tap water. This solution does not have to be made precisely. The instrument display prompts you to perform this bleach step at the beginning of the first run of each day. It is important to sterilize the instrument with bleach, as bacteria grow in the running buffer that remain in the bottom of the rig and tubing after the run. Bacteria secrete nucleases which break down the purified DNA. Now that you have set up your instrument, we'd like to review what we know about growing recombinant bacteria to obtain good plasmid DNA yields. The first suggestion is to grow bacteria in magnificent broth, or MR 2001. Make the broth by dissolving 25 grams of powder in 500 mils of pure water. We strongly suggest using nanopure, millicue, or double distilled water. Autoclave the broth for only 15 minutes. The second suggestion is to grow the bacteria for greater than 18 hours, preferably 20 hours, at 37 degrees centigrade in a gyratory shaker. The recommended shake rate is 200 to 230 RPM. Third, inoculate the culture with a fresh streak or colony of bacteria that is less than 2 weeks old. Glycerol culture stock is okay, but must be fresh. The age of bacteria should not matter theoretically, but it actually does. Never inoculate a liquid culture from another liquid culture. Fourth, we recommend that cultures be grown in one of 3 vessels. The first type of vessel is a Falcon 2059 or equivalent tube. This is a 17 by 100 millimeter tube with a snap cap that has an intermediate position for the cap to allow aeration. The second type of tube is a glass disposable 16 by 150 millimeter tube with an aeration type reusable cap. Third type of vessel is an Erlenmeyer flask. In either of these vessels, you must limit the volume of the culture to meet the volume of the flask. Limit the volume of a culture in the test tube to 2 mils or less, and limit the volume of culture in an Erlenmeyer to 1/10 of the total volume of the flask. The fifth suggestion is we recommend using high copy number plasmids as they work best with this machine. Plasmids in the range of 2 to 15 kb are in the ideal range. And finally, make certain that the culture is sterile. Wear gloves when inoculating and always grow with antibiotics. I will now describe the actual operation of the instrument. For this demonstration, I will use the 12-well cassette. The 24-well cassette will be explained later. For the first run of your instrument, we recommend that you use the test culture provided with your starter kit. Use of this culture will not only verify that the instrument is working properly, but it will allow you to become familiar with the instrument operation prior to using your own cultures.